So we were discussing about the IgM structure in the previous lecture. So I'll continue with the structure of the IgM molecule itself. So IgM is a pentameric having pentameric structure. So because of its pentameric structure with the 10 antigen binding sites, IgM molecule can bind to 10 small haptane molecule. Because of its steric hindrance, it can possible to bind only 5 haptanes. So IgM is also more efficient than IgG at activating the complement. Because of its large size, IgM does not diffuses well, therefore it is found very low concentrations in the intracellular tissue fluids. The presence of J chain allows IgM to bind to receptors on the secretory cells, which transport it across the epithelial linings to enter the external secretions that will be there on the mucosal surfaces. Now, we will see the next classification, immunoglobulin A or IgA. Although IgA constitute for about 10 to 15% of total immunoglobulins there in the serum, it is the predominant immunoglobulin class in external secretions such as breast milk, saliva, tears and mucus of the bronchial, urogenitary and digestive tracts. In serum, IgA exists primarily as a monomer, but polymeric forms also can be seen like dimers, trimers and some tetramers can be seen, but all containing J chain polypeptides. IgA of external secretions is called as secretory IgA, which is consisting of dimeric structure or tetrameric structure, also consisting of J chain polypeptide and a polypeptide chain called as secretory component. The J chain polypeptide in IgA is identical to that found in the pentameric IgM and serves the similar function in facilitating the polymerization of both serum IgA as well as secretory IgA. The secretory component, which is having 70,000 molecular weight, a polypeptide produced by the epithelial cells of mucous membrane. It consisting of 5 immunoglobulin like domains, and these domains will bind to the Fc region domains of the IgA dimer. IgA secreting plasma cells are concentrated along the mucous membrane surfaces, where secretory IgA serves an important effector function at mucous membrane surfaces, which are the main entry sites for most of the pathogenic organisms. Because of its polymeric, secretory IgA can cross-link large antigens with the multiple epitopes. So, secretory IgA has been shown to provide an important line of defense against many bacteria, example Salmonella, Vibrio cholerae and gonorrhea, and viruses like polio, influenza and retrovirus. So, breast milk contains secretory IgA and many other molecules that helps to protect the newborn against the infection during the first months of their life. So, the next classification is immunoglobulin E or IgE. The potent biological activity of IgE allowed it to be identified in the serum despite of its extremely low average serum concentration. It is 0.3 μg per ml. So, IgE antibodies mediate the immediate hypersensitivity reactions that are responsible for the symptoms of hay fever, asthma, hives and anaphylactic shock. So, IgE binds to Fc receptors on the membrane of blood basophils and tissue mast cells, where the cross linkage of receptor bound IgE molecules by antigen induces basophils and mast cells to translocate their granules to the plasma membrane and release their contents to the extracellular environment. This process is called as degranulation. As a result, a variety of pharmacologically active mediators are released and give rise to the allergic manifestations. This will lead to the allergic reactions in our body. The next last classification of antibody is immunoglobulin D or IgD. So, IgD has serum concentrations for about 30 μg per ml and constitute for about 0.2% of total immunoglobulin present in the serum. IgD together with IgM is a major membrane bound immunoglobulin expressed by mature B cells or nothing but plasma cells, and its role in physiology of B cell is under investigation, but till now no biological effector function has been identified for the IgD immunoglobulin molecule. Next we will see the diversity of antibody molecule. The very first thing is isotype. So, isotopic determinants are the constant region determinants that collectively define each heavy chain class and its subclass and each light chain class and its subclass within the species. So, each isotype is encoded by a separate constant region gene, and all the members of within the species carry the same constant region genes. So, within a species, each normal individual will express all isotypes in the serum. So, different species inherit different constant region gene and therefore express different isotypes. Therefore, when an antibody from one species is injected into the another species, the isotypic determinants will be recognized as foreign, inducing an antibody response to the isotypic determinants on the foreign antibody. So, the next is allotype. So, although all members of the species inherit the same set of isotype genes, multiple alleles exist for some of the genes. So, these alleles encode the subtle amino acid differences called as allotypic determinants that occur in some but not all members of the same species. The sum of individual allotypic determinants displayed by the antibody determinants is called as allotype. Antibody to allotypic determinants can be produced by injecting antibodies from one member of a species into another member of the same species who carries different allotypic determinants. Antibody to allotypic determinants sometimes produced by the mother during the pregnancy in response to a paternal allotypic determinants on the fetal immunoglobulin. So, the next is idiotype. The unique amino acid sequence of variable region of the heavy chain and variable region of the light chain domain of given antibody can function not only as an antigen binding site but also as a set of antigenic determinants. The idiotypic determinants arises from the sequence of the heavy and light chain variable regions. Each individual antigenic determinants of a variable region is referred as idiotype. In some cases, an idiotype may be actual antigen binding site. In some cases, an idiotype may be comprise variable region sequences outside the antigen binding site. Each antibody will present multiple idiotypes. The sum of individual idiotypes is called as idiotype of that particular antibody. Thank you.